This little video is going to be summarising the process of oxidative phosphorylation. So, in summary, this is the formation of ATP by the addition of an inorganic phosphate to ADP in the presence of oxygen. Oxygen is acting as the final electron acceptor in this process. So where does oxidative phosphorylation take place? This is a very simple diagram showing a typical mitochondria, and what you can see is there is an outer membrane and there is an inner membrane. Now, oxidative phosphorylation takes place on the inner membrane of the mitochondria. Now, within a mitochondria, there is a fluid-filled area known as the matrix, and to the outside is obviously the cytoplasm of the cell. So, just to summarise there, oxidative phosphorylation takes place on the inner membrane. Now, you might notice that the inner membrane of the mitochondria is also folded into these projections called cristae, and that increases the surface area so that you can have a greater number of electron carriers and a greater number of ATP synthase molecules, which we'll get to later. So how does oxidative phosphorylation actually take place? This little diagram summarises what's going on within the membranes of the mitochondria. So what we've actually got is, to the outside here, we have the outer mitochondrial membrane. Okay, so that is a phospholipid bilayer making up there the outer membrane. So out here will be the cytoplasm of the cell. To the inside, here is our inner mitochondrial membrane, and again you can see it's made up of a layer of phospholipids. Now, in order for oxidative phosphorylation to take place, a really important molecule, or really important molecules, are the electron carriers, which are embedded into that inner mitochondrial membrane. So I'm just going to draw these in very simply. One there, one there, one more there, and one there. Now, within the inner mitochondrial membrane, there are 4 types of electron carriers involved in this process: complex 1, complex 2, complex 3, and complex 4. Now, for the A-level OCR specification, you do not need to know the names of all these electron carriers. However, it does mention the name of complex 1, which is, get this one right, NADH coenzyme Q reductase. So that's the basic structure of those electron carriers. Now, as well as those electron carriers, embedded into the inner mitochondrial membrane is a molecule or many molecules of ATP synthase, which I'll just draw over here. Now, attached to the ATP, or making up part of the ATP synthase, is the stalked particle. Now, I'm not very artistic, so you'll just have to go with that. So there you go, that is the ATP synthase molecule. And here you can see, very important, I've drawn a channel, which is obviously associated with that stalked particle, through which hydrogen ions or protons can move. So, one other really important aspect of the basic structure of the mitochondrial membranes is this area in the middle. Now, this is known as the intermembrane space. Now, the the intermembrane space is really important throughout this process, as it allows what's known as a proton gradient to be established from across the intermembrane space to the matrix. So the matrix is actually the area here. So this will make up the matrix. So how does the process of oxidative phosphorylation actually take place? Now that we've got an idea about the structures involved with the process, we can start having a look at how the process actually takes place. Now, we know that we've generated many hydrogen atoms through the previous stages of respiration. And we've got lots and lots and lots of these hydrogen atoms which have been collected up and carried by the coenzymes NAD and FAD, which have been reduced. So we've actually got lots of molecules of reduced NAD and reduced FAD, which are able to transport hydrogen atoms to the inner mitochondrial membrane. Now, when they reach the inner mitochondrial membrane, those molecules, those coenzymes, reduced NAD and reduced FAD, will actually release those hydrogen atoms, which will actually break down or dissociate into hydrogen ions and electrons. Okay? Now that happens in the process of an enzyme which you don't need to know the name of. Okay, so the hydrogen atoms are split into their protons or hydrogen ions and their electrons. Okay. Now, I'm just going to follow the fate of the electrons to start off with. So these electrons are taken up by the electron carriers. To start off with, NADH coenzyme Q reductase. And then they move through the series of electron carriers. We call this the electron transport chain. You can imagine these little electrons moving, this pair of electrons moving through from electron carrier to electron carrier. Now, as they move through, they release energy. Now, that energy is used to pump hydrogen ions into the intermembrane space. Okay? And that will happen through complex 1, 3, and 4. Okay? So what we create is a very high concentration of protons or hydrogen ions within the intermembrane space. Okay? What you've created, therefore, is what's known as a proton gradient. So there is a greater concentration of protons within the intermembrane space compared to within the matrix. Now, those protons will then flow through the channel protein associated here with the stalk particle in ATP synthase. Just label that. And they will move down their proton gradient. We sometimes also call this an electrochemical gradient, okay, or a pH gradient. This will obviously be an area of higher acidity than inside the matrix. So those protons will flow through this channel protein associated here with the ATP synthase, and as they flow, they produce what's known as a proton motive force. Now, that proton motive force allows a molecule of ADP to combine with an inorganic phosphate ion and produce ATP. The vast majority of ATP is produced during this process, okay, and you're going to generate a lot of ATP by this method. Now, there's one thing that we haven't considered yet, which is what is the role of oxygen in this process. This process is known as oxidative phosphorylation. So what does oxygen do? Now, we've generated here lots of electrons as they've moved through the electron transport chain. Now, those electrons are going to re-enter the matrix. We've also got lots of protons moving down their concentration gradient and back into the matrix. Okay, and they are going to combine with oxygen to yield water. So oxygen is acting as, there's a little oxygen, I've got a half O2 here just so that it's a balanced equation there. So the oxygen is acting as the final electron acceptor. Without oxygen, this process cannot occur. Just one thing I wanted to emphasize as well. The process by which the protons move through the channel protein associated with ATP synthase and ATP is generated by this proton motive force. We call that process chemiosmosis. And it also occurs during the light-dependent reactions of photosynthesis, so it's worth bearing that in mind.